Welcome to the Robert Jenkins Microscopy Facility at the University of Wyoming. Our mission is to assist researchers in their microscopic imaging needs and to enhance student learning experiences. Students and faculty members from all across campus come to our lab to capture images of everything from animal organs to cells to viruses. Located in the Biological Sciences Building, our facility has both optical and electron microscopes, which can reveal macro to nanoscale structures. Let's start the tour with optical microscopes. Our first stop is the Zeiss Zoom microscope. This is a stereo dissecting microscope. With a large field of view and a 16x optical zoom, you can see structures at the cellular level. It has a motorized stage and is equipped with both bright field and fluorescent illumination techniques. It also has an optome, which is a structured device that allows you to make optical sectioning. That is, slice your sample optically rather than physically. We are now in the home of the laser scanning confocal microscopes. We have a Zeiss 700 model, an upright microscope that works well with fixed samples. It has 4 lasers, ranging from 405 nanometer to 633 nanometer. The way a confocal microscope works is that a small aperture or opening is placed in front of the detector. The aperture blocks out of focus light, therefore making the image sharp and crisp. It also gives the ability for optical sectioning. You can image the same sample at different focal levels and then build a 3-dimensional structure. You can also take multiple images to build a high-resolution capture of a larger sample, like a panoramic picture. For example, we can use this method to capture an entire mouse brain. Our second laser scanning confocal microscope is the Zeiss 980. It is an inverted microscope that is capable of imaging live cells. It contains 5 different laser lines, 3 fluorescence detectors, and 1 transmitted light detector. This microscope is also equipped with a super-resolution module that uses an array of detectors to offer higher speeds and more sensitivity to produce a higher resolution image compared to a regular confocal microscope. The images of confocal microscopes are fantastic, but if you need to image a lot of similar slides, it becomes very time-consuming. The slide scanner, on the other hand, is specifically designed for going through several samples with high-throughput imaging. The scanner can load up to 100 slides and process them in a convenient process. The scanner will automatically recognize samples on each slide, and tile scans the sample to stitch together to form a very detailed single image. Now we are in the 2-photon microscope room. This form of microscopy uses 2 lower energy photons, instead of just 1, to hit the same fluorophore simultaneously. A fluorophore is a chemical compound that can re-emit light to help make a specific point in the sample stand out for the viewer. This is like having 2 people instead of 1 pushing a vehicle. Each person will need only half of the energy. The advantage of 2-photon microscopy is that the lower energy photons cause less damages to the cell and penetrate deeper into the tissue. Our 2-photon microscope is specifically designed for in vivo imaging of live animals. While the animal is awake and active, you can image its neuron activities and correlate the activity with, for example, the movement of the pupil. Now let's move on to electron microscopes. As the name indicated, these microscopes use electrons instead of light as the illumination source. This can produce an even higher resolution than your typical light microscopes. There are 2 types of electron microscopes: a scanning electron microscope, or SEM, and a transmission electron microscope, or TEM. In our facility, we have a Hitachi Tabletop SEM. It provides limited resolution but requires little sample preparation. The SEM can be used for viewing surface structures of various samples, such as insects or plants. For example, you can simply pick a pine needle, place it on the sample holder, and image it directly. While the SEM observes the surface structure, the TEM examines the internal structure of objects. The Hitachi H7650 TEM we have can reach an ultimate resolution of 0.2 nanometer, and the magnification can be as high as 600,000 times. To think in terms of scale, imagine if I am being magnified for only 300,000 times. When I set my feet in Laramie and lie down, my body will cover the entire state of Wyoming, and I can use Grand Teton as my pillow. With such a high resolution and magnification, we can easily see nanoparticles, viruses, or a subcellular structure like mitochondria. Thank you for taking the time to tour the Robert Jenkins Microscopy Facility here at the University of Wyoming. For more information about what we offer and how you can reserve a microscope, visit our website at uwyo.edu/microscopy or email us at microscopy@uwyo.edu.